Deep in Pharmaceuticals in association with Higher Secondary Principals Forum.
Greetings students, I'm teacher Roqueinha Monteiro Pereira coming from Father Agner Higher Secondary School, Verna. And I'll be taking the topic for y'all, Microbes in Human Welfare. Chapter number 10 on your textbook. Okay, students, before I start with my lecture, I would like you to sit with your textbook and a pencil in your hand, because I'll be making you all mark important points on the textbook, both for the Goa board as well as for the medical entrance exam, the NEET examination, which many of you are aspiring. Okay, so please, the chapter number 10, just see the page number, it will be page number 171, okay, the chapter 10. And now, first of all, write on that chapter the marks for the Goa board. The marks for the chapter for the Goa board is 6. 6 marks chapter this will be for the Goa board. So you have to answer questions of 6 marks for the Goa board. And for NEET examination, there have been at least 2 to 3 questions asked. It depends, because now for NEET there are 90 questions, you all know that, from all the 11 chapters which you all are going to study. You all are having 16 chapters, of that 5 are deleted by the Goa board, but we have to see how the NTA decides. That is up to you all. I'll be teaching you all for the Goa board topic only, that is for 6 marks. Alright? Now students, when you talk about microbes, you know, first thing comes in your mind, microbes are pathogens. Okay? They cause a number of diseases, not only in human beings, but in plants and animals as well. And the important thing which is going on today is the COVID virus. Now this COVID virus is a microbe, it's a pathogen. It has taken over the world and has put us in this situation. So now how are the microbes? The microbes are always pathogens, they are always producing diseases. It's not so. So in this chapter, we are going to study the importance of microbes for the human welfare. So to start with the chapter, first let us study the important part. On the first page itself, it is there for you all. You can keep your textbook open, the first page open, to mark certain things. Now here, microbes are present everywhere. All of you know it. In the 11th standard also, you all have studied it, and biological classification. The second chapter in your 11th standard on your textbook. Okay, so the microbes are present in the soil, in the water, right? They are present almost everywhere. They are present in the soil, in the water, then they are present in our bodies, in the plants. Okay. All right, so they are present in our bodies, in plants as well as animals. Now, they are present where no life exists. That is in geysers where we have bath every day, in the thermal vents which are 100 degrees centigrade, deep in the soil, in the layers of snow, acidic environments, etc. This is there in your textbook, the same thing I have taken from the textbook. Now, this is very important. Microbes include not only bacteria and viruses. You might think that the microbes are present they are only in the form of bacteria and viruses. It's not so. The microbes include the protozoa. Protozoa, example I can take amoeba, then paramecium, which I've studied last year. Then the fungi. Fungi also trichoderma, the fungus, which I have studied, that also is a microbe. Then plant viruses, different ones, which we are going to study in biotechnology. And I'm also going to deal with that. Then viroids, the viruses. And then prions. Microbes include the prions. Now this is there in your textbook. In the second, fifth line, it is there. I want you to mark on this on your textbook with your pencil. Now, what is the meaning of prions? Prions are proteinaceous infectious agents. Prions are proteinaceous infectious agents. Now, this question has been asked in one of the medical entrance exams. So, you underline that with your pencil and write there 1 mark. The other things are simple, which you know that they are found everywhere. Now, bacteria and fungi grow on nutrient media. They go on nutrient media, which I am going to show you certain nutrient medium they are growing in the culture in the laboratories. Okay, the nutrient medium can be agar-agar, which they commonly use in the laboratories. Now, and they form colonies. These microbes, actually you know well, they are called as microbes because they are to be seen under the microscope. Okay, but when these microbes, the bacteria especially, the bacteria, virus are very difficult to be seen with the naked eye, but the bacteria, when they grow in the nutrient medium, no, they will form colonies, and these colonies can be seen with the naked eyes. Okay, so this is just the introduction which I have given you for your topic, microbes in human welfare. The first page, the important thing you mark that you know very well that they are found everywhere. But the common thing which you will not remember is a prions. Please mark there. I would like to show you this slide. This page, this is there in your textbook itself. I have just taken this picture from your textbook itself. Okay, I just want you to go have a look at this picture. Now the Goa board, no students, we have never asked you any questions based on this. Okay, but in the medical entrance exam, there have been questions on this. So you just have a look and mark certain things. Now, for example, first I will take this bacteriophage. Bacteriophage you will have studied in the 11th standard. Then if you have already studied the molecular basis of inheritance, there have been number of experiments performed on this bacteriophage. Okay, now in this bacteriophage, no, students, I want you to know the labeling of this. Now you can see on this picture, on your picture also, there are head, collar, tail, plate, and this here the rings are present, rings. Okay, so now here the rings, they might ask you to label in the medical entrance exam. Goa board, we have never asked you any questions, but they might ask you the labeling along with the questions. Okay, so I want you to know, remember this. Those are going to answer medical entrance only for them. Now here you can see the rod-shaped bacteria here. You can see the rod-shaped bacteria with a number of flagella here. Alright, then you can see the viruses here, one picture. Now here, this is a culture plate where I told you the bacteria on a nutrient medium, they form colonies and can be seen with the naked eye. Now this I am going to show you in my next slide. Now here, this is very important, students, to understand the remaining part of the chapter as I am going forward. Now can you see this? This is a culture in a petri dish. Okay, now the nutrient medium is present there in the form of agar-agar, so some other nutrient they might use also, but mostly agar-agar. And can you see this bacteria forming colonies? Now see, they are streaks like, and then when they disintegrate, they look like this. And each of these look like this. Now see the bacterial culture here. Your bacteria are growing luxuriantly on the nutrient medium. This is just an introduction for you to understand. Now, let us start with our topic. A real topic now. Now, in the chapter, no students, you all are going to deal with the following things. You are going to study microbes in household products, which is there in your textbook also, microbes in industrial products, microbes in sewage treatment, microbes in production of biogas, microbes as biocontrol agents, and microbes as biofertilizers. Okay, so these are the 6 subtopics which we are going to study in this chapter. So pay attention when you are observing this lecture. or going to the lecture, please sit with a textbook and a pencil. Once again, I am reminding you. Okay, now we shall take the first one. Microbes in household products. Okay, do you know, students, we are using the microbes every day in our life? Every day, including they are also present, you know, in our digestive system. So we go through one by one. Yes, now the first one which we are going to deal today is the production of curds. You all have eaten curds or yogurt. We prepare yogurt or curds at home. So I have just put a picture for you to see the curds, not the pudina leaves. The pudina leaves is just to when we present the dish. Okay, pudina is not a part of the microbes or the curd. This is just a presentation. So you are having curds at home. Some of you like curds, some of you don't like. But do you know the importance of curds? Curds is very important for our stomach, for our digestion. Now what does the curd, how the curd is obtained from the milk? We make curds from the milk, and what happens? How the milk gets coagulated into curds. Now there are lactobacillus bacteria present. Now these lactobacillus bacteria and others, they are commonly called as lactic acid bacteria, or in short they are called as LAB, and they convert the milk to curds. Okay, can you find it on your textbook, students? Can you just find it on the textbook? Yes. Now curds is produced by the LAB. Now what happens, you know, this lactobacilli, they produce an acid, and that acid, you know, converts the protein in the milk. Now what is the protein in the milk? Protein in the milk is casein. You all must have done some projects also in the 11th standard on this coagulation of proteins and different nutrients or different type of you must have used to check or to find out the casein. Same way you must have checked the casein. Many times students do these projects in the 11th, 12th, like in Goa Dairy, in Amul milk or some other Aditya milk, how much content of this casein or protein is present, or what is the effect of this protein on the digestion. So this casein or the protein is converted, is coagulated by this bacteria by producing an acid, and it becomes curd. Now what is the, this here I have said, now what happens here? The LAB or lactic acid bacteria coagulate and partially digest the milk protein. Some people cannot digest the milk protein. You know very well, students, we do not have an enzyme when we grow up. We do not have an enzyme renin. Renin is present in the infants, okay, which converts or digests the proteins from the milk. So as we grow up, it is not present. So some people have that acidity problem because of this milk and all. So these people can have partially digested milk in the form of the curds. Okay. Now, how is this curd produced? All of you know. A small amount of curd is added to fresh milk as inoculum. Inoculum. This can be a neat question. Okay, inoculum. What is inoculum? Inoculum contains millions of LAB. I'll be referring as LAB only, that is lactic acid bacteria. So now, a little of inoculum, say a teaspoon for half liter of milk, little warm, will be put in the milk and kept for some time aside, maybe for 2 to 3 hours, and finally the milk curdles. All of you know that, all of you must have seen curds, and your mothers also must be making curds in the kitchen. Understood, no? So what is inoculum? Inoculum contains millions of the bacteria. Alright? Now, the nutritional value of milk is improved by production of vitamin B12. Now, this is very, very important. The nutritional quality of milk is improved now by the production of vitamin B12. This question has been asked in the NEET examination. Earlier in earlier there used to be AIPMT, when there used to be a Goa CET and different COMED exams and other exams. That one there used to be AIPMT, which included the 11th standard as well as 12th standard portion. And this was also asked in the Goa CET examination. Okay. Now what does the lactic acid bacteria do? LAB does. They improve the nutritional quality of milk by production of vitamin B12. Please underline this in your textbook. Please underline this. Now, what happens by this LAB? Now, these LAB, you know, check disease causing microbes in our stomach. Okay, so they check the disease producing microbes in our stomach. So, this is very important. Mark there on your textbook, and outside the textbook on your left hand side, no, there is place. Okay, right there, number one, microbes in household products is production of curds. Put a hyphen and write down LAB. Remaining thing you can mark inside the textbook, because in the Goa board they have asked a question of 2 marks earlier. Give a few microbes, running 4 microbes which are used daily in a household products. So your number 1 is production of curd. Alright, and this is what is there. The next slide, second use. Yes. Now here students, since you all eat bread every day, you all eat dosa, idlis, you know that very well. Now, dough for dosa and idli. Dough for dosa and idli is prepared by the fermentation of the substrate. That is, for dosa and idli we use the rice as a substrate. Okay. And in that, you know, that is fermented to give a puffed up appearance, or puffed up fluffy influence or puffy appearance because of the fermentation by bacteria. Now you might wonder how this happens. Now whenever we prepare dosa and idlis, no, we use urad dal for that. Urad dal. Okay. Now urad dal, depending upon the consistency, is soaked in the water. So when the urad dal is soaked in the water, the bacteria get activated. Okay, urad dal is a source of bacterium. When it is soaked, the bacteria gets activated. Okay, and this bacteria ferment the dough, and this gives a puffed up appearance of dough, and that is because of the production of carbon dioxide gas. Now how that puffed up appearance comes, you know? Because the bacteria which got activated by soaking the urad dal, and when you mix it with the rice, the substrate, the bacteria become active and they give out carbon dioxide. Okay, and this carbon dioxide, you know, gives like holes like in the idlis. Holes. This is the batter. I put a picture of the batter which you must be seeing every day at home when your mamas prepare that at home. Okay, dosas and idlis. Same way, can you see this, students? In the bread also you can see this hole like appearance there and the puffiness in the bread. Okay, okay, so second one is, first one is dough for dosas and idlis, which I have already shown you. Dough for dosa and idlis. Okay, second dough is for the bread. Dough for bread is fermented using baker's yeast. Now this is very important. For the dosa and idli, you'll use fermentation by bacteria in the form of urad dal. It is not, the urad dal is not there in your textbook, but you'll know it, but just write it. And you might wonder from where it got activated. So as I've taken, since it is not there in the textbook, I have taken the source from the, I've googled it. So Google says, you can also Google it. Google says that in inactivated form, they are present in the urad dal and they get activated after they are soaked. Okay, now in bread. Next one is dough for bread. Second one, you put the point there. Dough for bread, no, is fermented by using baker's yeast. Please remember that. Now what is this baker's yeast? Baker's yeast is Saccharomyces cerevisiae. Baker's yeast is scientifically the bacteria Saccharomyces cerevisiae. It is there on your textbook. Please underline, students. The third one, dough for bread, and in that put a hyphen and put there baker's yeast. Mark it on your textbook with the pencil on the left hand side this page, page number 172 if you have not opened, and underline this also, Saccharomyces cerevisiae. Now this is very important. You can see this puffiness, this is puffiness and holes like. Now this holes like are produced, because of the release of carbon dioxide by the activated bacteria. Okay? Now look here. This is how they make the dough when making bread. Can you see this? Now can you see these, students? All these holes like are present there. Now here the bacteria, the baker's yeast, the bacteria, they are getting activated here. And when they start respiring, they give up carbon dioxide. As they give up carbon dioxide, the dough starts coming up. Can you see the dough? Now here I am showing you the baker's yeast. The baker's yeast or Saccharomyces cerevisiae is a scientific name. Now at home your mamas must be doing it. If they make cake and all, they use this baker's yeast or something they might be using. Your mamas must be making bread also at home. So they use this baker. The baker's yeast looks something like that. Something like this. Okay, in the laboratory also you all must have seen this is a practical in the 12th standard. Unfortunately this year the practical is cancelled, so you will not be mounting it. Okay. After soaking it in the water, the yeast looks like this, or the Saccharomyces, the yeast. This is the yeast here. Here the yeast, Saccharomyces cerevisiae looks like this. Can you see? Here is the yeast. Alright, and the yeast is budding over here. Can you see the yeast is budding over here? It is producing buds. Now this budding in yeast was a practical for you for the 12th standard earlier. Now since because of this COVID situation, it has been deleted from your syllabus. So you won't be observing it under the microscope. But 11th standard, if I am not mistaken, I think it is there, but the mounting will directly show a slide. Otherwise you had to mount it yourself. Okay? Now, students, you all know very well the third thing which you are doing is the Todi. You all know about Todi. We Goa, we produce Todi. We get Todi and we use Todi for fermentation to produce alcohol, and a very important one which we do in our household. We don't ferment liquor in the kitchen, but what we are studying is microbes in household products. So we produce sannas. So Hindu students might not know what are sannas, but you must have eaten them along with your Catholic friends. Our festivals are incomplete without producing these sannas. Okay, so what are these? How are these sannas produced? Now, Todi is used for fermentation of the rice. Okay, we soak the rice. Rice is a substrate, and we mix the rice with required amount of this Todi. Okay, and that gets fermented. Now, and then we produce the fluffy sannas here. Todi looks something like this. You shall have not. Now what is Todi? Todi is nothing, students. Todi is obtained from the palms. You know very well that Todi is obtained, from the coconut trees. But in some other states, Todi is obtained from some other palms also. Okay. Now this Todi is a traditional drink which is given in your textbook also. Todi is a traditional drink obtained from the palms, and it is used to prepare fermented drinks. Okay, traditional fermented drinks. Alright, and in the kitchen we also make sannas by using this Todi. Understood? No? So please write there outside your text, Todi is a traditional drink, because this question has been asked in the examination. So you should keep in mind. The next one is fermented fruits. Okay, please underline on your textbook fermented foods also and write it outside also, because by the time when you are preparing for your board examination, for your medical examination, you might not have time to go through all this. So if you write outside your textbook itself, you will remember for the revision. Okay, if you write and prepare notes in your notebooks, no students, sometimes you lose the pages, you lose the notebooks. So in biology, one thing about the Goa board, we ask questions totally from the textbooks. Totally from the textbooks, and we stick to our textbook to ask you questions. Now in the medical examination, there might be some additional things like from the CBSE, but not always. It is one and the same. Okay, that is the other part of it, because right now I am lecturing for the Goa board, and something here and there I will tell you the questions which have been asked in the medical entrance exams. The next one are the fermented foods. What type of fermented foods? Microbes are used to ferment fish, soya bean and bamboo shoots to make food. Now in your textbook, no students, they have given that much only, like ferment fish, soya bean and bamboo. I would take the example of your household. What we do? We ferment foods at home with the salt. We make pickles. Okay, we make paras, we make pickles at home, and you know very well what you add to that. You add salt to that, you add oil to that, and we take and we preserve all this by the method of fermentation. Okay, so I hope you all have got it. The next one. Now this, I hope your mouth doesn't water, students, to see the cheese on your screen. Here, cheese. Next one is the cheese. Now for the cheese, no, you write there on your textbook at the side, you know, cheese. And I want you to make 2 arrows, up and down, because we are going to deal with 2 types of cheese. One is Swiss cheese. Write an arrow on top and write on top as Swiss cheese. Alright. Now, how is this cheese produced? The texture, flavor and taste are dependent on the type of microbe used in production. The flavor of the cheese, no, and the texture of the cheese comes depending upon the microbes which are used. Okay, and accordingly we are going to study in your topic today 2 types of cheese. Swiss cheese. Now the Swiss cheese is produced, how it is identified. It has large holes, and these large holes are due to the production of carbon dioxide by the bacteria Propionibacterium shermani. Okay. Now Propionibacterium shermani. Now in the Goa board we have asked about this Swiss cheese. The large holes are produced by the carbon dioxide. Now students, you don't have to remember this big word Propionibacterium shermani. Okay, don't have to remember to write it, because it is difficult, the spelling, to remember. But for the medical entrance exams, they ask you in the MCQs, and they give you the alternate. Okay, and this question has already been asked in the Goa CET. It has already been asked in the Goa CET. So what was the question? The large holes in the Swiss cheese is because of the production of carbon dioxide. Once the question was like this, and another time in one of the medical entrance exam, the question was because of the bacterium Propionibacterium. Okay, please remember this is very, very important. So you write there itself in the form of arrow. And look at the cheese, students. Can you see the texture? Nice one, no? Very appetizing. Okay, so smooth cheese. And can you see the holes here? Very important is the holes are because of the production of the carbon dioxide. Okay, I hope you understood. Now, one more arrow. You have written cheese there, no? Cheese, and one arrow on top, Swiss cheese, and write the remaining things. Second one, you put an arrow and write this. So, second one is a Roquefort cheese. Okay, Roquefort cheese. Now Roquefort cheese looks like this and it is having all this black color all over. The holes are because of the release of carbon dioxide, and in the holes only you can see this black color here. Now this is ripened by growing specific fungi, Penicillium roqueforti. Okay, the fungi Penicillium roqueforti gives the special flavor for this Roquefort cheese. Okay. Now this was your microbes in the household products. I hope you all have written in the textbook and you all have understood this. After writing, you can go through once again, but don't forget students to make the notes outside the place which is there outside the textbook only is enough. Okay, write in the form of points. So 2-mark question. Mostly 2-mark question only will be asked from this part, if at all it is asked, out of 6 marks. So 1-mark question also is ready if you look at the text outside only, instead of reading the entire thing, and it will be a revision for you for your medical entrance exams also. The next one is microbes in industrial products, the next one. On the right hand side of your textbook it is there, microbes in industrial products. Alright. Now, look here carefully, you can underline afterwards on a textbook also. Beverages, that is the drinks, beverages like wine, beer, etc. and antibiotics. And antibiotics, you know what are antibiotics? Okay, you apply the penicillin medicine and all that, different type of antibiotics. They are produced with the help of microbes. So industrial products, what we are studying in this topic, one is the beverages and other one are the antibiotics. Please remember this. Okay, now these are produced in the industries on large scale, you know, by using large number of microbes. You cannot use few as you are using in the household products. Like that cheese also we don't produce in the house, but it is a household product. That's why cheese is included in that particular topic. Okay. Now, these microbes, when they are used in the industries, you know, they are grown in vessels. And these vessels are called as fermenters. Please remember these words. Okay. It is there in your textbook, underline, and you can write it at the side of your textbook also. On your right hand side, you can write it over there. Okay. So, in the industries when microbes are produced in large numbers, you know, they are produced in fermenters. Now see, student, this picture is there on your textbook. It is not very clear. I have put it a clear picture for you from the net. Okay. Now here, this is a single fermenter or a vessel. In this vessel, in the fermenter, the microbes are grown for fermentation, and here this is how the process or distillation or fermentation is going on, which we shall do later. Now, what happens in this fermented beverages? What happens over here? Now, yeast. Yeast is used here for the fermentation. Okay. Now, yeast is what? Saccharomyces cerevisiae, which you already know. That is used for bread making, is commonly called beaver's yeast. Please underline this and write outside your textbook also. In making bread, what we said? Baker's yeast. Okay. But here in fermentation, you know, it is also used and it is called as brewer's yeast because we are using it for brewing of beverages. Okay. And is used in fermenting malted cereals. Now, what are these malted cereals? No, I will tell you in the later part of my topic. Okay. Malted cereals and fruit juices. So, to prepare beverages, no, malted cereals and fruit juices are used. And what happens on fermentation of this? Malted cereals and fruit juices, that is important. What is produced is alcohol, ethanol. Ethanol, which is alcohol, is produced. Once again, 1-mark question can be asked in the Goa board. And this question has already been asked in the medical entrance exam. Please underline. And here I have given you an equation which is not there in the textbook, but in chemistry you must have studied it. Okay, I can't recollect if it is there in the textbook, but it is there. Here I put it for you. Now, these fruit juices and cereals, they contain glucose. This glucose, when it gets fermented by the yeast, it produces ethanol, the alcohol, and it releases carbon dioxide. Now, I want to tell you something. You all might not be drinking beer because you are too young to drink beer and all, but I'm just saying. You must have seen somebody open a beer can or opening a beer bottle. The moment the beer bottle is opened, first everything comes out if you shake the bottle. Or if you open slowly, you can see the holes coming out and this fizziness is coming out. Even in your soft drink, the fizz is there. Freeze, okay? The fizz or whatever you say, fizz is there. That is because of the release of carbon dioxide, which is trapped inside. The carbon dioxide which is produced by the respiration or by the yeast. Okay? By the yeast. Yeast respires to produce the carbon dioxide. Okay? Anaerobic respiration, which we shall study later. So that this like fizz what is coming out is because of the production of carbon dioxide. Okay? Now here students, I am showing you some alcoholic drinks on your screen. Please don't mind. I just wanted to put these pictures for you. It is just mentioned in your textbook, but I just thought I will show some pictures for you. Wine and beer. Okay, now how they are produced, alcoholic drinks which are produced, depend on the raw material. Raw material means in the form of cereals or in the form of fruit juices. Okay, now here I would like to tell you about the cereals. What is the cereal? Now cereal grain like rice, wheat or maize or corns. Alcohol is produced from rice, from corns also. Okay, these are the cereals. Now these cereals, are germinated by soaking them overnight. They slightly sprouted. The cereals, are slightly sprouted. The grains are slightly sprouted. And when they are sprouted, they are heat dried. Okay, to stop the germination. They are heat dried to stop the germination. Okay, and that is called as malting. It is not there in your textbook. You can make a note of it. The cereals are sprouted. Okay, by soaking. Cereals are always sprouted, grains are always sprouted by soaking. Okay, overnight or so. And then they are heat dried. Why they are heat dried? To stop the germination. Okay, and this process is called as malting. Okay, now this is some additional I have told you. Now alcoholic drinks depend upon the raw material used for fermentation. Then they are fermented and how they are processed. There are two processes. Alcohol is extracted only by fermentation and alcohol is also extracted by distillation. Alcohol when they are produced by distillation, they become strong alcohols. Okay, now wine and beer is produced from fruit juices and malted cereals respectively. That is wine is produced from fruit juices. Here is the wine. I have shown you the wine with a lovely glass over there and nice appetizing wine over there. Okay, so it is produced, student, from the fruit juices. Your parents might sometimes make this wine. They produce wine at home from the grapes or from some other fruits. Okay, I don't know much about it because I don't know how to brew the alcohol. I have never done it. Okay, so now from the fruit juices, by adding the yeast Saccharomyces cerevisiae, the alcohol is fermented. Okay, it is fermented to produce the wine. Now next is the beer. Beer is produced from malted cereals. Beer is produced from malted cereals. Please remember that. What is malted cereals? I have already told you. There is no time to do it again. I have already told you. Now, and one important thing is that wine and beer are produced without distillation. This is very, very important. It has already been asked in the Goa CET. So, wine and beer is produced without distillation. Alright. Now, here these are produced by the whiskey, rum and brandy. These are produced by distillation from fermented broth. Okay, this is very important. It is there in your textbook. Last part of the paragraph it is given. Whiskey, rum and brandy is produced by distillation from fermented broth. Now, I want you to write something over there on your textbook. It is not there in detail in your textbook, but I have taken it for you in detail. Whiskey. Whiskey is produced from malted grains or cereals. Okay, there is no time to explain to you about these grains and cereals again, but remember one thing. What is malting I have told you just for information, but it can be asked in the NEET examination, medical entrance. So, whiskey is produced from, remember this, from malted grains and cereals. Okay, then rum is produced from sugar molasses. It is given in your textbook. Rum is produced from sugar molasses. Please write this on your textbook. Sugar molasses, that is from the sugar cane. From the sugar cane, you know what are molasses while making sugar. The sugar cane is crushed and all, and the molasses are prepared. Okay, the next is brandy. Brandy is produced from wines or fermented juices. This is very, very important. Brandy, no, is later on, it is distilled from the wine. Brandy is prepared by distillation of the wine which is already prepared by fermentation or from other fermented juices. Okay. Now here, this is a distillation, this apparatus I have put it just like that for you, just to see distillation process in the lab. Here are the juices, then the heat is provided, and here the condensation takes place and the condensate comes out. Okay, I just put this for you if they're not there in this is how distillation takes place. Now next is the antibiotics. Okay, here are the meaning of antibiotic is anti is against and biotic is life. Now this is there in your textbook. Okay, now here I want you to underline chemical substances which are produced by some microbes that kill or retard the growth of other microbes. This definition you should know. Antibiotics, what are antibiotics? These are the chemicals which are produced by microbes, and these chemicals kill or retard the growth of other microbes. Alright, now here I am showing you a culture. This is a culture of the bacteria. Here the bacteria are growing on the nutrient medium, and these are the antibiotic disc. These are the antibiotic disc here. Okay. Now these bacteria which are present over here, these antibiotic has totally retarded the growth of the bacteria. Totally retarded the growth of the bacteria. And here still the bacteria are growing at the side, and here they are still growing. Okay. Now this was accidentally discovered by Alexander Fleming. It is there on your textbook. Alexander Fleming, when he was working with Staphylococci bacteria, he suddenly found that the growth of the bacteria is hampered like this. They are dying. Okay, when he was working with Penicillin notatum. So he started checking on that why it is happening. But his work was incomplete. Then the other scientists, worked on that, which I will show you later. Scientists like Ernest Chain and Howard Florey, these two scientists completed Alexander Fleming's work, and finally penicillin antibiotic was produced, which was used in World War II. Okay, so this is a disc where this is not there on your textbook. I have just put the disc for you to understand. Now one more thing I would like to, sometimes when there are infection, Pus is there, a swab is taken and it is sent in the lab for testing. And swab, it's tested like this by putting all these antibiotics to check for which antibiotic the bacteria are resistant. And then a proper antibiotic is given for the curing of the wound. Okay, whenever there is a curing of the wound, a specific antibiotic is given instead of just trial and error method. So it is taken and then a culture is made and the different types of antibiotic discs are used, and then it is seen in which antibiotic the total growth of the bacteria will be retarded. Okay. Now the next slide. Now this I just want you to mark on the textbook, students. Alexander Fleming worked on Staphylococci when he was Staphylococcus bacteria. I already told you that, and he gave the name for this mold as penicillin. After the mold penicillin notatum. Okay. Now he found that in the bacteria, the Staphylococci bacteria, the mold, the fungus grew. Okay. And this fungus name he gave as Penicillin notatum. Okay. It's a fungus, it's a mold. Okay. So Penicillin notatum, he gave the name. The Staphylococci were the bacteria which he was working on. So suddenly a mold appeared in his petri dish and he gave the name as penicillin. After the mold, penicillin notatum. Later on, effective antibiotic was established by Ernest Chain and Howard Florey, and all of them were awarded a Nobel Prize. This is there on the textbook, you can just mark it. Now, penicillin was extensively used in World War II to treat American soldiers. This also is there on your textbook, you can just underline. Second thing, you can underline this. Antibiotics are used to treat diseases like plague, whooping cough, diphtheria, leprosy, etc. Now about all this plague, whooping cough, diphtheria and leprosy, you all must have already studied in human diseases. Okay, the other chapter in human disease, if you have not studied, you will be studying about it. Another teacher will be dealing with this with you in human disease. In detail, you all are going to study about it. Now here students, I just try to put the picture of this scientist, Howard Florey. This is Alexander Fleming who started first the work, and Florey and Ernest Boris Chain, they completed the work. Alright. Now here students, penicillin culture looks something like this. This is the dish, petri dish, where the nutrient is there, and here the bacteria are growing. And look, this bacteria secrete a secretion here. Can you see this? This is the antibiotic. Can you see this secretion? Small transparent globules like, can you see? Can you see these globules like? Yes. So this is the secretion by the mold. And this is known as penicillin. This is later on processed and all in biotechnology. You all are going to study about it once again, how the processing and packing and everything is done and sold in the market. I just thought I will put this slide for you. Alright, this one, it is very clearly seen. Alright, because it is not there in your textbook. The next one, the last part of my lecture is the chemicals, enzymes and other bioactive molecules. It is there on your textbook once again. Now this chemicals, enzymes and other bioactive molecules, I have put in the form of a chart here. Students, I want you to copy this chart. Okay, we are studying three: chemicals, enzymes and bioactive molecules. Three things. I put in three different colors. Now, what type of chemicals are produced? Chemicals like citric acid, acetic acid, butyric acid, lactic and ethanol are produced. Lactic acid and ethanol you all have already studied. I have already taught you now. Lactic acid in the curds, ethanol in the alcohol. Okay. Now, these products, or these chemicals are produced by these organisms. Aspergillus niger is producing citric acid. Acetobacter aceti is producing acetic acid. Clostridium botulinum is producing butyric acid. Lactobacillus is producing lactic acid, and yeast Saccharomyces cerevisiae is producing ethanol. I want you to write this because Goa board also they have asked this question. For 1 mark also this question has been asked. You should know which organism is producing which chemical. Okay, now here enzymes produced from the microbes are also producing enzymes here. The enzymes lipases is used in detergents to remove oily stains. Okay, the oily stains which you get on your clothes and all, no, they can be removed by detergents by using lipases. So you see in the TV ads and all that stain removing, oily stain, stubborn stain are removed. Now those stains can be removed by using the lipases, and these are produced by the microbe. Same way pectinases and proteinases, these are the enzymes, you know, which are produced by the microbes for the clarification of bottled juices. See, when we make juices at home, no, we have to strain them with the strainer because the turbidity is there. But when you get juices from the market, you know, they are very clear. Now, why are the juices clear? They are clear because they use the enzymes pectinases and proteinases to clear the enzyme. Now, the next one is Streptococcus. Now, Streptococcus, you know, produces the enzyme streptokinase. You know, Streptococci is a bacteria. Strepto, you know what it is, in a line it forms a chain. The cocci bacteria, the circular, cocci means circular bacteria, they form a chain. That's why they are called as strepto. Streptococci bacteria, you know, they produce an enzyme streptokinase. Now, these are used for clot busters in myocardial infarction. Okay, now what is myocardial infarction? Myo is the muscles of the heart, myocardial muscles of the heart, and infarction is formation of a clot there. Now what happens now? The coronary artery supplies blood to the heart muscles. Sometimes because of atherosclerosis. Now what is atherosclerosis? Thinning of the blood vessels due to the deposit of fats in the blood vessels. Now this atherosclerosis, no, causes thinning of narrowing of the blood vessels, and when the blood is passing, you know, a clot might be formed, and that clot, you know, will lead to heart attacks. Okay, now whenever a person gets a heart attack and all, they use the streptokinase if it is because of the clot. Okay, now if the blood vessels, if the person has undergone a heart attack and there is a clot in the blood vessels, that clot is bursted or broken down by using the enzyme streptokinase. Please underline it on the textbook. Okay, I hope you all have understood. Last year I have studied about heart attacks. Heart attack, heart stroke, cardiac arrest. There's no time to explain to you all this. What is there this year only I'm explaining to you as a clot buster. Okay, now next is bioactive molecules. Bioactive molecules, two types we are doing. Trichoderma. Trichoderma is a fungus. Polysporium. Now from trichoderma, no, you get a bioactive molecule cyclosporin A. And the cyclosporin A is used as a immunosuppressive agent. Now what is the meaning of immunosuppressive agent? Whenever a patient undergoes a transplant, like kidney transplant, heart transplant or retina transplant, our own body recognizes its own self. This also you are going to study in human disease chapter. Okay, so our body recognizes our own, and the body, when it is not its own, tends to reject. So the immune system has to be suppressed so that the body doesn't reject the organ. And in this case, no, cyclosporin A, okay, a bioactive molecule is used as an immunosuppressive agent. Okay, and the last one, a bioactive molecule is yeast. Now yeast here is of different type. We have been studying of Saccharomyces cerevisiae only, but here we are using Monascus papyrus. Monascus papyrus produces a bioactive molecule statins, and these statins, you know, is used for blood cholesterol lowering. Now you know about blood cholesterol. You ever heard about cholesterol, which is a fat deposit which is present in the blood? It is useful also, but if it is too much, like there are different type, HDL, LDL, okay. So bad cholesterol, good cholesterol is say. So the bad cholesterol, when it is there in higher quantity, what happens, you know, it gets deposited in the blood vessels and leads to other heart attacks and other complications which you have just done. So to lower or keep the cholesterol under control, no, statins are used. You will get different type of statin, atorvastatins, 5 mg, 10 mg. So all these tablets, you know, are made by using the bioactive molecule which is produced from the yeast. Okay. So now this is very, very important. Copy the chart. And second thing, this question has been asked in the board exams at least 2 times for 3 marks. From this part, you know, all the 3, streptococci, streptokinase has been asked for 1 mark also, MCQ also, streptokinase, cyclosporine, statins, all these are asked for 3 marks in the Goa board as a question. Okay. Apart from that, 1 mark question has also been asked in the Goa board examination. This is very, very important. Please do not skip this. You have to study this, and it is very important for MCQ. On this, you know, clarification of bottled juices, an application type of question has also been asked in the Goa board. Then detergents to remove oily stones in the Goa board as well as in the medical entrance exam. Okay. Thank you, students. The remaining part of the chapter we will do in the next lecture.
Prudent Scholars, powered by Lupin Pharmaceuticals.
अब आपकी सुरक्षा आपके हाथों में। ल्यूपी सेफ हैंड सैनिटाइजर, आपका और चेन्नई सुपर किंग्स का फर्स्ट लाइन ऑफ डिफेंस। अब आपकी सुरक्षा आपके हाथों में। ल्यूपी सेफ हैंड सैनिटाइजर, आपका और चेन्नई सुपर किंग्स का फर्स्ट लाइन ऑफ डिफेंस। अब आपकी सुरक्षा आपके हाथों में। ल्यूपी सेफ हैंड सैनिटाइजर, आपका और चेन्नई सुपर किंग्स का फर्स्ट लाइन ऑफ डिफेंस।